everyone. For those of you who do not know me yet, I am Dr. Lauren Schmidt, the new Chief Science Officer at the Phelan-McDermid Syndrome Foundation. I'm so excited for you guys to be here for our webinar on the natural history study. We are very lucky to have three of our very well-known providers today. We have Dr. Alex Kolevzon, we have Dr. Liz Berry-Kravis, and we have Dr. Sid Srivastava. I'm sorry if I butchered those names, but we are so excited. So how today's going to run is I'm just going to give a quick little intro, deal with some housekeeping things, and then I will turn it over to those three. At the very end, we have some of our colleagues or our collaborators from Jaguar Therapy on, and they'll answer some questions about the downward extension of the natural history study. And then we should have plenty of time for question and answers. We were collating some of those questions ahead of time, so we'll have those. And then any questions that come in, please use the Q&A box. So again, thank you so much for being here. We are very excited to have our speakers. The natural history study has been funded twice now by the NIH. We are in this interim period waiting to hear about the third cycle. At the foundation, we have been able to help continue to collect data. And thank you to our donors who have provided those funds so that we could continue collecting data for the natural history study. And we've also been able to provide some additional travel to the sites for that. And then Jaguar Gene Therapy will be able to talk about the travel that they're also able to offer for the downward extension. I don't think I have anything else to say at this point, so I will leave it for the three doctors to take over.
All right. I'll do the broad overview. So this is going back, I think, 12 years now. We, three of us, along with two other sites, collaborated to start a natural history study in Phelan-McDermid syndrome, where our goals were to characterize the clinical syndrome as broadly as we possibly could and to track the natural history over time. And we did this really to establish clinical trial readiness for when the time came that we'd actually have really meaningful therapies to test. When you look at the natural history, it's really important when we're designing a trial to figure out what kinds of outcomes we want to measure, what kinds of things change over time, what kinds of things stay relatively stable. And so this was a really critical first step. The second thing we wanted to do is to develop biological markers, either using things like imaging or EEG measures, to see if we could define ways of predicting treatment response or tracking treatment response over time. And then the third thing we did was very clinical, that Sid led, to try to develop consensus guidelines that were kind of an improvement over the original guidelines we developed in 2014. And that's been now disseminated and completed. So we've got consensus guidelines that sort of help figure out how to assess and monitor kids over time and also make some recommendations in terms of treatment. Fast forward, we've now kind of submitted the third iteration of this grant. And these are 5-year grants, and we expect to be funded soon. And then I would say in that decade or so, there's been an enormous flurry of industry and biotech and academic interest in developing treatments in Phelan-McDermid syndrome. So we got quite lucky that people feel as if Phelan-McDermid syndrome is a kind of strategic opportunity to develop new treatments for developmental disability and for autism in general. And I guess Jaguar was one of the first industry or biotech partners that came into the space and decided they were going to try to go after gene therapy. And then in that context, and I know I'm talking a lot, I'll stop in a minute. In that context, we added a new natural history study that was going to start as young as 12 months. I think the original natural history started at 3 years, and then we scaled it down to 18 months, and now with Jaguar down to 12 months. And with Jaguar in particular, it's really important to have a natural history data set because we're not going to be using like a placebo control. Obviously, with the gene therapy, that probably isn't an ethical thing to do. So the natural history of the syndrome without gene therapy intervention can serve as something of a control over time So I know that it's been very, very helpful to Jaguar in terms of their regulatory submission and their preparations for the clinical trial. So now I guess we are talking about the details of the natural history study, and I'm going to stop talking. Maybe Sid or Liz can add something that I forgot.
No, I think that's just a really key point, that having genetic therapies, natural history is really, really, really important. And because it gives us the trajectory of the normal disease and allows us to understand whether the genetic treatment has actually impacted the disease. And in some cases, especially if you follow the patient along and then they're in the natural history study and then they start treatment, you can actually look at intra-individual change, where you can look at, does the patient, how fast were they developing in certain areas, and what do they do now after they had the therapy. So this is a really kind of powerful thing, for sure.
One thing I just would definitely add is that it's really a testament to the community at large, the families, the individuals, the foundation as well, to really spearhead collecting, allowing this data to be collected, that has led to this point, this really important point where we are able to investigate precision medicine, gene therapy options, and other options. So I think it's a strong testament to the community at large, and a very exciting time to be in the PMS community.
So yeah, we were just gonna kind of focus in on the natural history study and like what findings you guys have had so far. We have already had a question in and sort of like what's been published, what kind of things have we found out from the natural history study.
Oh yeah.
That would be great for you guys to highlight.
Sure. Well, let's see. I think we've published more than 25 papers among our group. We've published studies about the clinical features, about the biomarkers, like using things like visual evoked potentials and EEG in general. And we published consensus guidelines. So there's been an enormous amount of productive work that's come out of this. And in many ways, we've kind of launched a whole clinical field based on this natural history study. Let's see, I think probably, well, Sid, maybe you could talk about, to me, one of the most impactful papers that we published really relates to these consensus guidelines. Maybe you want to talk just briefly about that so that everyone's aware.
Sure, absolutely. So Alex previously published the first set of guidelines from 2014, was it?
Yeah.
Talking about different features to look for, to evaluate for in somebody with PMS, whether that's related to the neurological system, whether it's related to development and the heart and so forth. When Alex first published those guidelines, there was, compared to now, much less that was known at that time. And the interval, there's been tremendous wealth of knowledge that we've gained collectively from published studies, including from the natural history study. So what we did is, together, we leveraged this consortium that we've built and grown to study natural history. We assembled different sort of groups focusing on development, focusing on neurology, focusing on cardiology, other aspects, other medical aspects, and assigned group leaders to each of those areas. And we basically reviewed updates in our knowledge, updates in the literature, and tried to come up with updated practices, best practices related to each of those different areas. And what we tried to do is have family representation involved in each of these different groups. And so it's not just clinical and scientific input, but also family input too, which I think was a really important aspect of things. We published these guidelines, I want to say two years ago or so, which are available through the foundation, through the website. And I think represents, culmination of the natural history data, culmination of prior publications and scientific knowledge, and it's impactful because it allows clinicians who are taking care of patients with Phelan-McDermid syndrome to better provide care for the families. And so I think all of us are collectively, proud and happy to have been a part of that process.
Yeah, I think the other thing we've spent a lot of time doing is figuring out which kinds of measures whether it's a parent report or a cognitive measure or a language-based measure, are relevant to Phelan-McDermid syndrome and which kinds of measures we should be using in clinical trials. Liz has spent a lifetime, at least my lifetime, working on different types of outcome measures in Fragile X syndrome and various other rare genetic disorders. So, Liz, maybe you can talk a little bit about the new instrument we developed specifically for Phelan-McDermid syndrome.
Okay. Yeah. So standardized measure, we've explored a lot of standardized measures in the natural history study, and it is important to have some data on those. But a lot of times in developmental disorders, the standardized measures were not created for those disorders, so they don't necessarily match up to the way the patients perform, and they don't necessarily detect change the way we would like. And for instance, we've looked at whether some standardized measures like the Bailey, which is a developmental test, and the Vineland, which is that endless interview that everyone hates to do, about how many developmental skills your child has and how they function in life. So those measures, we know, unfortunately, miss a lot of important changes. And we've studied that actually in the Angelman trial and found out that the standardized measures only detect about 20 or 30% of what families feel is a really meaningful change for the child. So as a result, we need to have other measures that are more granular and can detect important changes in diseases like PMS. So we've been working with Neuron to develop a CGI, which is a clinician global impression, where you just rate, is the patient improving, kind of the way you would in clinic, where you ask a lot of questions and take information from all sources and decide, is the person really better? Have they gained something new? Are they behaving better or sleeping better or whatever? And so we've spent a lot of time validating that. Neuron used it in their initial phase 2 trial and showed that it could detect change. We've showed that it has good intra-rater reliability. In other words, if I rate it one time, I'm going to get roughly the same thing the next time. We've done fidelity so that we can show that different raters would get the same rating on it. And now we've done interviews of clinicians to make sure it's really relevant to PMS. And some of this was things that the FDA requires to have a valid measure. But it really looks like this measure is pretty specific to PMS. It interrogates the domains of PMS that were important in the registry study, the PMSF questionnaire, as well as in the disease concept model that Kirshank did. So hopefully this measure will help us with being able to accurately determine when changes have occurred that are important, and then we can use the standardized measures as a backup, with the hope that we can see change on both types of measures, but that we're not missing anything.
Mm-hmm. Yeah, and I'll just highlight how meaningful it's been to be able to share that measure, including with Jaguar. So this is something that Neuron developed for their trial, but has every intention to be able to share so that future trials can also be able to use it. And we're of course going to use it in the Jaguar study. The third sort of area that I'll talk about just briefly, and then I'm seeing some questions pop up specifically about the gene therapy trial, so we'll inevitably get to that. But the third area we've been working a lot about is more kind of objective measures. So it's obviously very valuable to observe children. It's very valuable to use standardized measures, as Liz discussed. It's very valuable to get feedback from parents. But all of these things are also subjected to a lot of bias, right? We all want the kids to get better. Obviously, you all want your kids to get better. But things like EEG or imaging, for example, are totally agnostic, right? And these measure things like electrical activity of the brain. You can give people different stimuli, like visual stimuli, and you can see response in the brain, and you can measure the extent to which there's like excitation, basically, and the ability to kind of absorb information. You can look at just sort of a resting state and the balance between excitation and inhibition. So there's all kinds of ways of measuring electrical activity that we think might be able to help us determine who responds to treatment and who responds to treatment early on, perhaps even before you start to see later changes in terms of like language or cognition, which of course is our main goal. So these are things that we're going to be using in clinical trials going forward, including in the Jaguar trial. let's see. There was a question about the age, which I think is a really important question. And the question related to whether or not there's any real evidence that younger children may respond to treatment better than older children, for example, or adults. This is a question that gets asked all the time, and we actually don't have clear evidence of that in the case of Phelan-McDermid syndrome. But we do know that, of course, the brain is more plastic in earlier stages of development, and as you age, it becomes less plastic, less able to kind of make changes in response to learning. I mean, that's true for all of us. It's certainly true for me. But that's the rationale behind the very first patients that are going to be enrolled in the Jaguar Gene Therapy study are going to be the kind of the younger patients. So, we're focusing now on 2 or 3. We'll hopefully be kind of creeping up to older kids, 6, 7, 8, 9 eventually. And then ideally, this will be a treatment that applies to everybody. And I do think that there is evidence, at least based on preclinical models using mice, where you can manipulate when you turn genes on and off, that indeed, if you turn the SHANK3 gene on later in development, including in sort of the equivalent of adulthood, you can still see changes in the clinical features of the syndrome. So we expect that a gene therapy, for example, or really any treatment, can potentially have an impact on people with Phelan-McDermid syndrome. Well, we would just hypothesize that impact would be greater in younger kids, and that's that's the main rationale for starting in younger kids.
Absolutely.
And there's another easy question to answer, which is, can we know preliminary data and outcome of the child who received the gene therapy a few months ago? So we can say, at this point, it's been about 4 months, that this child is doing well, that there's been no significant safety concerns, but it's really way too early to make any real comments about the benefit, the clinical efficacy. It's just too preliminary. And so we're not going to be really discussing that. It's just way too early days, but we can definitely say with confidence that they're doing well, tolerating it, and there haven't been any significant adverse events.
Thanks for sharing. And we have we collected a bunch of questions online previously, so I do want to go back to those in a second. But since you did bring up the younger age range, this might be a good opportunity from either from Dan or Gina from Jaguar Gene Therapy to come on and kind of talk about the downward extension of the natural history study and how that both relates to what has already been collected, but then is a little bit separate than what is potentially planned in the third iteration of it. So, Dan, I'll hand it over to you.
Sure, happy to. Good evening, everyone. So I'm Dan. I lead clinical development with Jaguar. And, maybe just to reiterate some of what we heard from our PIs, the natural history study that's been ongoing for the last, 10 years has been incredibly important for our development program. So, those data that have been collected across many patients have helped us understand what Phelan-McDermid syndrome looks like, has helped us as a company design a trial to evaluate how gene therapy may work, or if it may work in these patients. And so a lot of the outcomes that we're collecting in our clinical study mirror the outcomes that were collected in the natural history study. And so when we think about what it takes to get to approval, we want to be able to show that the treatment is effective, that there is some meaningful benefit that comes from receiving the gene therapy, and that it's safe. And so having natural history to compare that to say, here's what the trajectory of disease looks like, here is what development looks like in these individuals with PMS when they're not receiving gene therapy, but maybe they're receiving physical therapy, speech therapy, occupational therapy, ABA, a number of other interventions. This is what the natural course of disease looks like. That gives us a baseline to be able to compare what the potential benefit of our gene therapy is. So the data that's been collected to date has been incredibly informative. It gives us some ideas of things that we can look for. And for instance, Alex spoke about, Dr. Kolevzon spoke about the electrical activity in the brain that we can see. There's a very specific type of phenotype that we see in individuals with PMS. And so one of the questions we can ask, for instance, is, well, after we give the gene therapy, can we see evidence that we're changing the biology of the disease? And so having that natural history gives us an opportunity to compare patients who haven't received treatment to patients who are receiving treatment in our study. And then, of course, as we go longer, we want to be able to see, are individuals developing new skills? Are they reaching developmental milestones that they hadn't previously been able to reach? And similarly, by looking back at natural history, we could say, well, in our gene therapy study, it looks like perhaps somebody's reaching a milestone at a particular point in time. Is there evidence that we would expect something like that to happen in the natural history? Or does this speak to the benefit of gene therapy? So there's a lot of questions that we can ask like that having the natural history allows us to ask those questions and to evaluate the potential benefit of our treatment. So really just highlighting how important it is to collect these data. And we recognize it's a commitment. There's a lot that goes into a family traveling and giving their time to provide these data. But importantly, it helps us understand the natural history of PMS, and in our case as Jaguar, hopefully it helps us get to an approved treatment someday. But it'll also benefit any other companies that are looking to bring additional treatments to families affected by PMS. So in the case of the additional work we're supporting, as Alex mentioned, they've gone through several iterations of the DSC, the Developmental Synaptopathies Consortium, and hopefully are close to receiving funding for the latest iteration. One of the areas that we wanted to supplement was collecting data in individuals on the younger side. So I think historically the DSC had been open to individuals 18 months and older, and there's probably fewer individuals on the lower age, the lower part of that age scale. And so what we wanted to do was provide additional support to collect natural history outcomes in individuals 12 to 36 months of age, approximately. So we've been working with all three sites, Boston Children's, Mount Sinai, and Rush University, to be able to provide some supplemental support so that they could collect all of the outcomes that they collect in the DSC natural history. And as Lauren mentioned, there's also additional funding that we'll provide to support the travel to those sites. So I think where we're at right now is $1,400 in travel stipend that we provide to families. Actually, we provide funding to PMSF. PMSF actually provides that funding directly to families. But that's meant to help offset some of the costs associated with the travel to the sites. And again, as with all the data from the DSC, these data will be incredibly important in helping us define our path to eventually, hopefully, seeking approval for JAG201.
Mm-hmm. There was a specific question about how do we sign up for the natural history study, and no doubt Lauren and PMSF will post contact information for the sites. And I'll also say that although the current ongoing study is sponsored by Jaguar and focused on younger kids 12 to 36 months, we really expect to get our third iteration of the DSD funded, in which case we're going to be able to open it up to additional families who haven't been participating of all ages. So there'll be lots of opportunities, we expect.
Yeah, I think that's a really important clarification that kind of right now, and then in terms of the natural history study, in terms of new enrollees, it's really limited to that younger age range, which I can post the information that we have on our website. And then there are still some follow-up studies from the DSC2, or the second iteration of natural history study. And then fingers crossed, hopefully we'll get a bunch of new enrollees once NIH comes through, hopefully with funding, fingers crossed. I think there's been some questions that we got before, as well as now, kind of around more of some of these biomarkers, whether they're in blood, EEG, MRI. Is there anything that we've found from the natural history study that is exciting, that we think we could move more into the clinical trial phase as an outcome measure, a secondary outcome measure?
Yes, I think there are a number of promising measures. One specific example is something called a visual evoked potential that was developed in part by Paige Syper, who's the chief psychologist at the Sierra Autism Center. When you look at a checkerboard where there's a contrast between white and black, you basically receive a signal to the visual part of the brain that produces a very specific waveform. And you can show virtually anybody one of these contrast reversing checkerboards, and they'll show the same waveform. And it's got trough and then a peak. And the trough and the peak correspond to excitatory activity of the brain and reflect the ability essentially of nerve cells to sort of absorb information and conduct information. And it's sort of one of the fundamental underlying processes of learning and memory. And then the peak that follows is the inhibitory response, essentially. Because Phelan-McDermid syndrome, you're missing your SHANK3 gene, that protein is an anchoring protein for these glutamatergic synapses. And so what you see in Phelan-McDermid syndrome is a real suppression of that initial trough that reflects the excitatory or the glutamatergic activity. And so we think that this is a way of potentially predicting treatment response and tracking treatment over time, so that we would expect that as symptoms improve and as nerve cells are supported by additional SHANK3, particularly in the gene therapy study, we should see an enhancement or an increase in the amplitude of that initial trough, suggesting more powerful excitatory activity, which is again critical for learning and memory. This is at the moment based on very small early pilot data in like an IGF-1 trial, basically. But in terms of like proof of principle and promise and kind of scientific merit, we're very optimistic about it. But it's still at this point, pretty exploratory. I don't know if Dan spent a lot of time thinking about VEPs, and Dan spent a lot of time looking at VEP waveforms. Maybe you want to add something.
I'm just trying to go back to the question, but yeah, no, I think that's right. So, I think VEP is an area that's of interest to us. There's a very similar or related measure, which would be AEP, so auditory evoked potential. And essentially, we do believe that these would be the earliest areas to look for indication of change, because the threshold for, measuring a difference is, something that should appear earlier, right? We're basically asking, is there evidence in the electrical activity that there's a change in response to a stimulus? So with VEP, it's that visual stimulus, the contrast reversing checkerboard. With AEP, it's a sound at a specific frequency, and it produces, again, similar electrical activity. And so our thinking is all we have to do is change the biology to be able to see that change reflected on an EEG, which is the measure of that electrical activity in the brain. And then to Alex's point, the hope is that if we're changing that, that's showing that we can change how the brain responds to a stimulus. It's showing that perhaps there's an increased ability to interpret that stimulus, and then that would be foundational for learning. So longer term, potentially that would predict benefit on different types of outcomes.
Great. Thank you for that. And some of the previous questions that we've gotten about the natural history study is also just sort of trying to better understand what is included and not included, kind of in terms of what we've learned about PMS medically. And so like anything about autonomic dysfunction specifically came up in terms of, how many patients are experiencing autonomic dysfunction. Is there anything else, from, the medical side, clinical consensus guidelines that can be mentioned about autonomic dysfunction?
Well, we do have some of the questions on the clinician forum address, various kinds of autonomic function, but I don't really think we have a lot of detail on that yet. That maybe is an area that we could delve into more.
Yeah, I mean, so the natural history study does a pretty comprehensive overview of all kinds of medical features. And if kids had autonomic dysfunction, we would be able to capture it, but not in an especially objective way, other than doing our sort of basic physical exam and neurological exam. In general, much of it is sort of qualitative. We're kind of asking families about their experiences, asking about things like, thermoregulation, for example. We're asking about things that are sometimes very hard to measure, actually, but it's very descriptive, and it certainly helps us understand the phenotype. And then the ideal would be, once we realize this is an area that's relatively common, we would actually dig into it a little bit more in depth and start to think about, offshoot studies that would be much more specific. And so we're thinking now about things that are really much more common. So sensory sensitivities, sleep problems, for example, Obviously, epilepsy is a common problem. So things that we can really think about more carefully in terms of targeting in the context of clinical trials: language impairment, cognition, activities of daily living, motor skills, things that are sort of broadly applicable to the entire group. And then I think eventually, if we start to see other changes that are specific to a subset, we would start to spin off studies that ideally would better understand that subset and then ideally develop treatments for that.
Great. Something that's also come up is sort of for families who are interested, either sort of in this downward extension for the 12 to 36 months or kind of the future iteration. What can families expect kind of when they show up, let's say, to Mount Sinai or Boston Children's? What does that experience look like? And how burdensome is it? How much do you help with things? What can they expect?
They should expect to have fun. I mean, these are great teams. These teams love working with kids. We love working with families. I mean, I'll let Sid and Liz answer, but I'll say just very briefly that we're very, very sensitive to the fact that kids can only tolerate a certain amount of testing over a certain period of time. So we're not going to torture your poor child or your adult. Well, I guess in this case, we're talking about little kids. So, we take breaks, we have snacks, we let kids run around. We're generally very, very accommodating. Usually visits take a few days for that reason, in fact, because we want to sort of pull the best performance we can out of kids, and we don't want to be biased because they're hungry or tired or fed up with us. Much of the evaluation actually occurs through the eyes of the parent. Right? We're doing a lot of parent interviews. Parents are doing a lot of filling out of forms. There's some obviously cognitive testing. There's some autism-focused testing that's directly with the child. But again, nothing would take more than a couple of hours, and we wouldn't be doing things for an extensive period of time, just out of sensitivity to the burden.
Yeah, I completely agree. And I think the two things I wanted to say is that, one, all of us kind of are of the mindset that we're really thankful and appreciative for participation in the study. I think we're grateful to learn from you, from the families, from the children. It's really, and that gratefulness, I think, extends to not just us, but the teams at each of our sites. And the second thing is that I think we try to be as flexible as we can with the sense of if there are, if some part of it can be done in person, if there's some parts that need to be done remotely or whatnot, we try to work with you because we completely understand it can be, it can be long. Sometimes it can be tight. Sometimes there are other family members or siblings there, and too, it can be long. So we really try to do our best to be as accommodating to you guys as possible.
Yeah, I think that's pretty much how all of us work, because we have many projects in other developmental disabilities and we have the same approach. We try to have toys, we try to have stuff available. Our autism center is very keyed into, they do the testing there for the PMS study, and they're very keyed into how to keep kids comfortable and give them breaks and keep them working, but not like overburdening them.
Mm-hmm. One thing I hear a lot is that families appreciate being able to sit down with doctors who've actually seen kids with Phelan-McDermid syndrome. So much of what we do in the evaluation is actually not so much evaluate, but just answer questions. I think people sort of value the perspective of someone who's seen dozens, even hundreds potentially of cases, and that I think families, they're giving a lot in terms of time and effort and travel and all that, but they're also, I think, getting a lot. And I should mention that sites also provide reports that summarize the assessment. Often those reports can come with recommendations. Those recommendations can be used to advocate for more services for your child, for behavioral therapies, for educational accommodations. There's a lot of value in these assessments.
Yeah, I think it's really important for families come in and they feel like no one really understands their condition all that well. And it just gives them a chance to answer all the questions, to get a feel for like where their child fits in the condition, whether symptoms they have are common in the condition, and even sometimes whether a thing that an associated symptom that they may have is actually due to the PMS or is just something else that's in the family. And I think it really helps to kind of sort out what's what. But also, most of the families that come to us really like to talk to me about what's the future of PMS, like what kind of treatments are we developing and how far away are we from having them and how do they work, and like they can't get that from their local doctors. So that's always like after we get done with the whole clinical assessment of the child, that's always a big discussion.
Everyone should go to Rush because Liz apparently has a crystal ball. I mean,
No,
not crystal
no,
ball,
no. We just talk about like what's in basic science that could actually move.
No, I understand. Okay.
I don't ever tell people exactly when it will move to people because
If only.
that
If only.
is very hard to predict.
Yeah. There's actually a question about that in the Q&A about sort of timeline. It's just so speculative at this point. It depends so heavily on what we learn from what, we've only had 2 patients enroll in the or I guess 3 in the gene therapy trial. So I think it's really very, very early days to speculate about how soon it will take before it's actually approved, and depends so heavily on safety and clinical efficacy and regulatory hurdles. There's a lot that goes into this. Dan will tell you that they've spent the last 6 years Shank 3 all day, all the time, and that's before they even got into the clinic. So I think it'll move faster than that going forward. Maybe Dan, you want to comment?
Yeah, I think maybe what would be helpful for families is to understand the timelines of our trial. So, to date, as we've shared, we've dosed 2 patients, one at Mount Sinai, one at Rush. Our goal for this first study is to dose a total of 6 patients, but we've got some room to dose a few additional potentially. As Alex said, I think our goal is to eventually get to treating a wider group of PMS patients, right? So while the first patients are young, on the younger side of the age scale, eventually our goal is to develop this treatment for all patients affected by PMS. So, we may have some opportunities to do that here within our first 6. I think, realistically, we'll dose our 6th patient, sometime next year, and then we'll be looking at the outcomes. So, looking at 6-month outcomes, looking at 1-year outcomes to understand what case can we make on the benefit that comes from treatment, and then what does that safety profile look like? And then the next step would be to have those discussions with the FDA, and based on the data that we've collected to that point, get some agreement on what a registrational phase study would look like. Likely include more patients, wider age group. I think there have been other questions about things like regression, deletion size, but individuals with different characteristics we'd be evaluating, and then that would be the study that would hopefully lead to an approval if the outcome was successful.
Thanks for clarifying. One of the things that came up previously is sort of wondering, what from the natural history study kind of in terms of the data has maybe changed how you do things as a clinician or a provider? Like, are there recommendations that have been coming out kind of from the data that are changing, what you do with patients?
Maybe I'll start, and then Liz, feel free to add. I think one aspect of things is some of the neuropsychiatric symptoms. I think we're, appreciating the prevalence of different psychiatric conditions within PMS, including conditions like catatonia, including other sort of aspects of neuropsychiatric decompensation, and learning about different things that can be the medications that can be used to target those type of symptoms within this condition, which can be a debilitating source, distressful to families, functionally impactful. So I think over time, having that knowledge or building that knowledge, which we will continue to build and expand, I think has been helpful. So that's one example that comes to mind. And Liz and Alex, feel free to add some more.
I think that having some of the measures that we've collected and having the trajectories of patients over time is really incredibly important. I think also understanding, the spectrum of how frequent is this symptom or how frequent is that symptom. Could we use seizures as an outcome measure? Well, no, not really, because we know that, only somewhere between 30 and 35% of patients have seizures. So understanding what is the percentage of patients that have one thing or another is really important. And it's important to have it in a cross-sectional kind of population, not, sometimes the populations you hear the most from are the people that maybe have the most problems, and you don't hear from families who are getting along pretty well and don't have all that many problems. But in a natural history study, the idea is that you're collecting everybody's information, so you really understand how frequent are different kinds of symptoms in a disease, and then you can use that to help with the trial design. But in the end, PMS is a cognitive disorder, and having background on the rates of developmental progression and how can we best measure that is just it's just a huge advance. It's huge for every single neurodevelopmental disorder that's looking at implementing treatment.
Absolutely. And I do want to make a quick plug that there will be a special issue in the American Journal of Intellectual Disability that will have a bunch of data from the natural history study. I think there's 4 or 5 papers that are coming out. So we will make sure that, we highlight those. I know we're going to have a blog post about it. And then both Dr. Audrey Thurman will be on to talk about it. So we'll get even more updates sort of once those are officially published. I did want to make a plug about that. Quick question that we've also gotten from a couple of people is, what about families who are outside the United States? Is it possible for them to participate in our natural history study? Have you had kind of people from either Europe or Canada or wherever participate? What does that look like?
So to date, we have not had people from outside the United States participate. And obviously, we're sensitive to the issues there, and there's many families that I'm sure would love to participate. But we've definitely made that a restriction, as have in addition to the English language speaking requirement. These are definitely shortfalls on our part and limitations, but that's the way it's been done up until now.
That's helpful. And I think especially the English language part, because a lot of the measures that you are using are only available in English or don't have the direct translation. So I think that's, some of those inherent limitations that sort of exist in the research world as well.
Yeah, I mean, the both the measures and also the clinicians,
Yes, that too. And then we've also gotten some questions, and I know this is something specifically you've been working on, Alex, in terms of what we've learned about kind of the mortality rate, and where are we with the mortality project?
Thanks for asking. Yeah, I think we're obviously very concerned and really motivated to learn a lot more about themes around the increased risk for mortality, the exact kind of prevalence, and we are asking families who have had losses to provide us with medical records so that we can really analyze them carefully and hopefully discover some specific themes and then start to think about potentially preventative measures. So at the moment, we have had, I think, 3 families submit medical records, and we recognize that this is kind of a big ask and often coming at a very sensitive time. And we were super grateful for the families that have participated. And we're hoping to collect information from about 10 families and then do some subsequent interviews. And then we would obviously disseminate our findings as quickly as possible. But at the moment, we're still in the kind of heavy data collection phase and recruitment, obviously.
Yeah. And thanks for sharing with that. And that is also something that if you are interested in participating, you can email us and we'll make sure we get you to the Mount Sinai group. And that's something that can be done virtually, remote.
Oh, yeah. Everything. Absolutely. Yeah. Absolutely.
Okay. Great. Some questions around, kind of going back to, I think you can probably see in the Q&A box, going back to there, around interventions that might be effective in reducing regression. I'm assuming maybe talking about neuropsychiatric illness. If you want to talk to that, since I know many of you on our consult group.
Yeah. Well, so as Sid mentioned, we've learned an enormous amount about the neuropsychiatric features. And I think that there's sort of two vulnerable periods for the risk of regression. One is early, before about 6 years old, and one is typically around puberty. And I think the regression that occurs around puberty is often coming in the context of new neuropsychiatric changes. So sometimes it starts with like very serious anxiety and OCD, like obsessive compulsive types of features. And then it can lead to mood changes where there's like lots of ups and downs in your mood. We've heard a lot about the risk of catatonia that can occur in that context. And so we've learned 2 or 3 really, really important things. So one are some medicines can make things significantly worse. So we recommend using things like selective serotonin reuptake inhibitors, medicines like Prozac, with extreme caution, and to use medicines like antipsychotics or like risperidone or Abilify with extreme caution. These things can definitely make this situation much worse. And on the flip side, if you intervene early enough, medicines that are considered to be mood stabilizers can be very, very effective. And medicines in the class of what's called benzodiazepines, which are anti-anxiety and they're a little bit sedating, can be extremely effective, can oftentimes prevent mood cycling, or if unfortunately things have started to really unfold and you even develop catatonia, they could certainly break that cycle if they're used properly and aggressively. And we've put a lot of recommendations both in SID's guidelines and on the PMSF website. And we have a medical advisory committee that's put out letters that could be used for the emergency room, or primary care doctors. So I think we've really learned a lot about this whole area, and we definitely have some clear recommendations that tend to be pretty effective.
I know we're closing up here, but I do want to get to this one question that kind of is focused on sort of the future of the natural history study, both in terms of like, let's say this next one, the 3rd round gets funded. Will there be additional sites? And then what does it look like 5 years from now? Is there going to be a 4th cycle, or, what do we do from there?
Definitely a 6th site, Jimmy Holder at Baylor, Texas Children's. He's 100% going to be part of our 6th site if we get funded. And definitely we are not going to have a 4th cycle because the NIH caps it at 3 cycles. But we will continue it in another way, either through industry partners. And hopefully by the time this 3rd iteration is finished, we will actually be clinical trial ready, and we'll be at the phase where there'll be lots of people that are testing new treatments. So it'll be a robust field with hopefully multiple options in the next 5 to 7 years.
And I think it's also important to remind the special issue I just talked about is on the 1st cycle of data. And so it also takes a while for a lot of this data to get analyzed and published. And so, yeah, there will be, hundreds of papers from the natural history studies.
From SID, hundreds of papers from SID.
Liz, you were going to add something.
Oh, I was just going to say, yes, it's 3 cycles, but after that, if we still need information and trajectories, the hope is that we could do something like the Angelman syndrome field has done, where they've established this sort of ABOM, which is Angelman biomarkers and outcome measures, and industries that want to move forward in the treatment with it for Angelman syndrome participate in this. And so actually right now, a group of industry contributors, as well as the Foundation for Angelman Syndrome Therapeutics, are funding the natural history study while we're in a hiatus. So I think being able to bring industry in as a funder to get data that might be needed going forward, even after 5 years, because drug development progresses slowly, and we might find we need things in the process of doing the initial trials. I think bringing industry in as a collaborator for that is really is a definite option.
I saw Dan shake his head yes, so we're gonna.
Can I just comment? I know. I mean, the sentiment was shared, right? But we're at Jaguar incredibly grateful for the families that contribute their time and their information to this data collection, and of course to the excellent sites. And Alex, Liz, and Sid on the call. A company like us would not be able to develop the treatment that we're developing today if the DSC didn't exist, if we didn't have all of the learnings from the DSC to help inform our study design. So it's incredibly important. And to Alex's point, more companies will come. Hopefully, more treatments will be developed, and natural history will continue to be important. So I'm optimistic that companies would, find a way to collaborate to sponsor ongoing research.
Absolutely. And the Phelan-McDermid Syndrome Foundation is so dedicated to the natural history study as well. And, we've obviously been able to, thanks to donations from probably many of you who are on the call right now, have been able to kind of fund things in the interim while we're waiting. And so I would also imagine that we will be very committed to making sure that research funds will be available to kind of continue, whether it's specific projects or, specific collection of data thereafter. So that is definitely our commitment to the natural history study as well. I know there's a lot of some additional questions coming in specifically about the Jaguar Gene Therapy trial, and I'm not I'm not trying to totally ignore them, but Dan and I and potentially some other people from Jaguar Gene Therapy will be having a session, whether it's a podcast or a webinar, that is more specifically focused on the gene therapy trial coming up. We're going to set a date and get information out. So save some of those questions for then so that we still have something to talk about. Because we want to make sure you guys definitely get to know the Jaguar team, just like how we have, and kind of build a relationship with them as they continue to do work in our community. Any final thoughts from our three doctors?
I think I'll start by saying that I'm optimistic about the future of PMS research. This optimism is driven by this incredibly passionate community that we have of the families of researchers. I'm so lucky to be part of this group with these amazing colleagues and this amazing consortium, and again, the families who really inspire us each and every day. I think it really makes us want to continue to do as much as we can to help support research and help improve and lead to better outcomes for our families.
Absolutely. I agree with everything Sid said. And it's really a partnership. We're in this in partnership with the families to bring everyone to better outcomes and improved futures for people with PMS. And any little steps we can do, like the natural history study, I think it's really a pleasure to work with all the families, and hopefully we'll be successful. I feel confident, and I feel like there are things going into the future. It's just going to take a little time.
Hmm. Yeah, well said. Nothing to add. Feel exactly the same way.
Well, we at the foundation are just also, grateful for the families who have participated and dedicated so much time and energy to providing this data that has been so pivotal for the field. Thankful for the, doctors on this call and the other sites that, are shepherding this forward. And, I know all three of you are sort of clinicians first and researchers second. And so I think that really our families really can feel that. And, thank you, Jaguar, for also sort of helping continue this extended downward for some really important data, not just for the gene therapy trial, but for our knowledge about development in general. So thank you, everyone. And finally, again, I just want to thank our donors. I know I might sound like a broken record, but we really wouldn't be able to kind of provide the travel funds to get to the sites or fund this interim period without the donations from you guys. So again, very incredibly grateful that money, goes towards really important things. So thank you. We will keep you updated on what we plan to do with the Jaguar Gene Therapy webinar, podcast, however it turns out. And then as more data comes out from the natural history study, we will definitely be posting that and keeping you guys in the loop. So thank you so much for being here, and stay tuned for hopefully some more exciting things in the near future.